think we can start. Good morning again, everybody, and thank you very much again for all of you that have been brave enough to get up earlier and come here at time. So thank you very much. Today I'm sharing this session. I'm Sara Bubesit. I'm a researcher at the Food Research Institute of Catalonia in Spain, but I'm also, as you could see in the opening ceremony, the re-elected secretary of the ICM Executive Board. And today it's my honor and also it's a very great pleasure to introduce our speaker, Paul Ross. He's the director of the APC Microbiome Ireland since 2019 and professor of microbiology at the University College Cork. Formerly, he was the head of the College of Science, Engineering and Food Science at the university, and before that, he was head of the food program at SIGASC. His main areas of research are in the antimicrobials and anti-infectives, in applied and also fundamental aspects. Also the gut microbes, the gut health, bacteriophages, and human, animal, and gut pathogens. He has won many awards, including the Doctor in Science in 2009, and was elected a member of the Royal Irish Academy, both in recognition of the research achievement. And today, Paul will talk to us about bacteriocins as antimicrobial tools to edit microbiomes for food and health applications. So the floor is yours. Thank you very much.
Thank you very much, Sarah. And good morning to you all, and thanks a million for getting up. I hope I worked it. Just to say, like many of you, I've been looking forward to this conference for the last 2 years. And I just want to thank the organising committee. I know you've been through a lot for organising such a fantastic conference. And just to say how much I've enjoyed it so far. And I was particularly interested to see that we had a number of microbiome talks, which makes my life a little bit easier, that I don't have to go that much into the microbiome. But just, I put up the slide, again, we saw it yesterday morning, but just to highlight the complexity of the microbiome. We work quite a lot on not just the bacterial component, but on the virome as well, which is an area that's gaining a lot of attraction. But I won't go into that this morning. But just to say we are all different from each other. Our microbiome is like a fingerprint. It's intrinsically linked to human health. And the microbiome, unlike the human genome, is malleable and we can change it. Age, diet, things like antibiotics, host genetics, shape our microbiome, but not really in a precise way. And one of the things we're trying to do at APC is come up with ways to manipulate the microbiome in a more precise way. And so how do we manipulate the microbiome more precisely? And one way is through antimicrobials. And we're all used to doing this through, say, antibiotics for animals, biopreservatives for food. But there is a great need for new antimicrobials, move away from chemical preservatives on the food side, need to improve shelf life. There's increased interest in natural fermentations. But on the medical side, there is an urgent need. And WHO came out there a couple of years ago, alarmingly saying that there's very few new antibiotics under development, and the current pipeline are simply modifications of previous ones. In 2019 alone, 5 million people died of antimicrobial resistance, and it's set to go to 10 million per year by 2030. And so, at APC Microbiome, this is roughly our thematic structure. So we're a microbiome centre of 300 people. It was set up in 2003. It's nearly 20 years old now. And it's set up around 4 research teams. And the research team I'm going to talk about is one that's headed up by Dawa van Sinderen, Colin Hill and myself. It's mainly Colin's and my work on antimicrobials. And basically it's in a theme called Microbes to Molecules, where we come up with new biotherapeutics, new pharmabiotics as we call them. These can be small molecules, bacteriocins, peptides, live biotherapeutics and so on. And I just want to say something about bacterial warfare within microbiomes. So microbiomes are a theatre of activity composed of trillions of organisms, all in competition with each other. They compete for nutrients, they cross-feed with each other. They produce a range of antimicrobial metabolites, including antimicrobial peptides called bacteriocins. Work that we're doing in the rumen at the moment would suggest that about 38% of the bacteria in the rumen have the genetic potential to produce bacteriocins. And in work we had done a good few years ago, we had suggested that bacteriocin production is a probiotic trait. And you see this in this study here, we used the probiotic Lactobacillus salivarius, and which basically, in this particular case, eliminated a listeria infection here. And if you genetically deleted the bacteriocin production, you still get the infection. And so showing that it's the actual bacteriocin production that's protecting the animal, the live animal in this particular case. Bacteriocins themselves, produced by gram-positive bacteria, fall into two main classes. They're basically modified or they're post-translationally modified or unmodified. And these class 1 bacteriocins, they can have quite unusual structures. Class 2 are normally just simple peptides. And an example would be niacin. Niacin, we're coming up to celebrate actually niacin's centenary very soon. It was discovered in 1928, a small peptide of 3.5 kilodaltons, and it has 5 rings in it, and it's extensively used in the food industry. And I just wanted to show you I suppose, to emphasize one thing about niacin and other antibiotics and these class 1 bacteriocins, that the bacteria go to quite a lot of trouble to make them. And they're quite complicated structures. And so you start out with a peptide. Sorry, what I do there. Okay, sorry. Start out with a peptide, and basically leaders, serines and threonines are dehydrated. They react with cysteines to form rings, and in all, niacin has 5 ring structure, and you have a complete pool of genes there in the cell that catalyze this. And so it's quite an unusual structure, and I say this for a reason that will become apparent in a while. And so I've built the talk around 3 kind of simple questions. One is, can the spectrum of activity of bacteriocins vary and be manipulated? Secondly, can bacteriocins be used to modulate the gut microbiota composition and functioning? And lastly, can bacteriocins be used to cause a physiological effect in an animal? And so if I take the first one, so bacteriocins themselves, and we have been isolating them now for decades. And here is just a list of some of the bacteriocins that we have, and their spectrum of inhibition, which is on the right there. And where you see a yellow box, it means it basically kills it. I suppose the message to take from that is bacteriocins can vary a lot in their spectrum of activity. And just to show you an example of work we done a few years ago on a bacteriocin that's produced by Bacillus thuringiensis called churicin, it basically kills a very narrow spectrum, Clostridia and other bacilli. It's an example of a class 1 bacteriocin, but not an antibiotic. It's called a sectibiotic. It has sulfur to alpha carbon rings on it that you see in the bottom left. And one nice thing about this is we can take advantage of churicin in that it doesn't really cause the collateral damage that other antibiotics do, such as metronidazole and vancomycin. And these are in human fecal fermentations where you see in the arrows here that Clostridium difficile is killed equally by churicin as well as metronidazole and vancomycin. But metronidazole and vancomycin completely change the microbiota. Churicin doesn't. One nice thing about bacteriocins as well is you can change them, you can make variants of them genetically. And we've literally made thousands of variants, because they're gene encoded, of niacin through self-cloning. Some of these variants, like M21V, has enhanced anti-listerial activity, they can have enhanced staphylococcal activity, enhanced diffusion, even more activity against gram-negative bacteria. And I suppose significantly what we have been able to do with these is we have, through the Environmental Protection Agency, which is our competent authority for EFSA, we've got approval to use these modified bacteriocins without GM labeling. The bugs themselves are GMs, but for contained use, the peptides aren't. And so the second question I wanted to answer is, can bacteriocins be used to modulate the gut microbiome composition and functioning? And we had done a small study about a year ago, where we were looking at the effect of niacin in these fecal fermentations, like I've just shown you, for churosine. And what we found was that niacin, in a dose-response manner, can modify the microbiome in these fecal fermentations. And you can see it there, where you get a relative increase in proteobacteria and a decrease in firmicutes and actinobacteria, particularly bifidobacteria, which are extremely sensitive to niacin. And this prompted us to do a trial more recently in vivo in pigs. And so what we were thinking here was that now if we encapsulate bacteriocins such as niacin for intestinal release, would we be able to modify the gut bacteria? And so in this particular case, we had 4 groups. This is N is equal to 10 pigs, and this is just post-wean pigs. We had niacin powder. This is purified niacin, an encapsulated niacin. This is with ethyl acetate. And we had we also did the encapsulation material. And basically, what we did here was we treated them with niacin for 3 consecutive days, and then we had a follow-up on 3 days post-treatment and 10 days post-treatment to see what happened with them. First of all, we were very surprised. Like a dogma in bacteriocin research is bacteriocins are broken down in the intestine, and so they couldn't cause an effect on the microbiota, which is why we encapsulated them. But what we found was that when we looked at the fecal samples of these pigs, we found in the niacin-treated group that in fact we were able to get antimicrobial activity of them while they were being treated. This is at T24, T48, and T72. And even 3 days post-treatment, we could see some of the activity. What we also see is that we see the peptides in the mass spec of intact niacin. And so this was a big surprise to us, that niacin survived gastric transit in this case and wasn't broken down by chymotrypsin and trypsin. And this just summarizes a number of studies that we did just in vitro, where we actually synthesized niacin without the actual post-translational modifications and just tested them against chymotrypsin and trypsin. And what we found is that the actual post-translational modifications seem to protect niacin from proteolytic breakdown. And so one thing was, if niacin's getting through, was it affecting the microbiome? And so what we see here is, this is in the middle box, what you see, this is at T72, which is at the 3rd day of feeding. And you see that here, the alpha diversity is really decreased in both the niacin encapsulated and the niacin powder, not in the encapsulant or the control. What we also see is that the niacin-treated group at 72 hours all start to cluster together. And so the microbiome of niacin-treated groups is very different. And these are Sankey plots that follow the actual trial, where you look at the microbiome over a number of days. And what we see here is a reduction in gram-positive bacteria. Remember, niacin is principally active against gram-positives, and a reduction in firmicutes and actinobacteria. In particular, as I said, the bifidobacteria are very, very sensitive to niacin, with an increase in proteobacteria. And also, niacin seemed to reshape the firmicutes. In particular, there was a reshaping of the firmicutes with increases in negativicutase and a decrease in bacillus species. In addition, there was a large increase, or relative increase, in gamma proteobacteria. I suppose this shows that not only did niacin survive, but it had an effect on the microbiome. It also affected the functionality of the microbiome. So we looked at it, this is just by HPLC here, looking at the effect on short-chain fatty acids. And they were lowered with niacin treatment. And you see at T72 there, you're getting about a 40% reduction in acetate, 66% reduction in butyrate. And that somewhat recovers in post-treatment. When we did shotgun analysis on all these samples, sequencing, and we see that also the pathways for some short-chain fatty acids were reduced. Pathways for butyrate, and I just point them out there, was reduced with niacin and substituted with those from gram-negative bacteria. And you see in the bottom, it's just a incidence of genes for acetate, butyrate, and propionate synthesis. And what the dark ones are, the dark kind of purple ones are gram-positive, and the lighter ones are the light pink ones are gram-negative. And so you say there, for butyrate, the main butyrate producers seem to be gram-negative. And so this in vitro trial with niacin showed us that niacin can survive passage through the mammalian intestine, is relatively resistant to intestinal proteases, causes a reversible change in the community, changing gram-positives to a gram-negative type predominance, and also causes a change in microbiome functionality. And again, on the right-hand side, these are the top 20 species that are affected with their, and you see mainly gram-negatives that, but not exclusively, that go down. Some of them actually, you know, like Ruminococcus andoriae, go up. Also remember that niacin can be engineered to modulate the spectrum, and so has potential in humans and foods. And so we feel that what this trial is showing us is a potential by isolating maybe new bacteriocins with different spectra that are antibiotics, or by engineering niacin, that you would be able to change microbiomes in a particular way, almost like editing them. The last thing I want to talk about is, can bacteriocins be used to cause a physiological effect? And again, this is very recent unpublished data that we have. And I want to introduce a project that's headed up by Catherine, who's here, Catherine Stanton, called MetLab. And it's not from Breaking Bad, but it's methane reduction using lactic acid bacteria. So methane is the second most significant contributor to greenhouse gas emissions in Ireland. In fact, you can hardly switch on the radio at the moment, but they're on about it all the time. And Ireland has a target of 25% lower greenhouse gas emissions for its agricultural sector by 2030, a target that a lot of us are worried about because it's very difficult to get it. And also, since quotas were abolished in 2015, Ireland has been one of the fastest growing agricultural areas. But now there's pressure on livestock to reduce numbers, or reduce emissions at least. And so what we wanted to do was we wanted to exploit bacteriocin-producer cultures to target methane-producing bacteria. And so this is a collaboration that is between Ag Research, INRA and Wageningen, and Teagasc and University College Cork. And so we have done a large-scale screening against Brevibacter, looking at inhibition of them, looking at hundreds of bacteriocin-producing cultures, and have, with our colleagues in Ag Research, developed a large bank of strains. But I'm just going to show you two of them here. And this is not just inhibition of the strains, but also inhibition of methane production. And this is by two cultures, a Lactobacillus plantarum here. There's two cultures on this, you see, and they reduce methane output by this strain. And also, in this particular case, it's actually Nisoplan is what we're using. And you see it kind of flatlines methane production by the strain. But the two cultures we're using, one is the nisin producer, and one, Lactobacillus plantarum, is a plantaricin producer. They actually, I think the nisin producer inhibits the plantarum as well, but didn't really cause us a problem in what I'm going to talk about. And so our idea here was, we knew these cultures inhibit Metana brevibacter, and so we decided, and these were trials that were done in 2020 that we're still analysing. So we made animal feed, 50 tons of it, and you see them there spreading the culture on the actual, spreading the cultures on the silage. And cows consumed quite a large amount of like 14 to 15 kgs per day, and they contained about 10 to the 11 to 10 to the 12 CFU per day. So they're getting quite a very large dose of these strains. The trial itself was basically to compare the effect of feeding grass silage inoculated with lactic acid bacteria and uninoculated grass silage on animal productivity and methane emissions. There's 30 cattle involved, 2 15s, and it was a 28-day feeding trial. And so you had one group that had the culture and one group that didn't. Actually, by the bales were purple, the ones that had the culture. And also, we measured basically production traits in the animals, including milk production. They were housed indoors for this. We measured methane emissions using this green-fed system, where the animals can just go in there as they want, and it measures methane emissions. And as I said, this was a co-inoculated silage. And these were the results that we've, just recently got. And what we found that was, it was run for 7 weeks, and between week 4 and week 7, we were getting a 5.88% reduction in methane production by the animals. And you can see there, the p-values after 4 weeks. And so the outcome of this trial, so we had bacteriocin strains that inhibit methane production by Metana brevibacter. The silage fed could be manufactured containing high levels of live strains. Cows indoor fed with the strains dropped in methane production by 6% from week 4. There was no impact on weight gain, milk yield, or other production traits in the animals. And we had some of these animals that didn't participate in the trial that were on this feeding regime. They were cannulated. And so we've ongoing shotgun sequencing and so on these animals at the moment. But I just put, I suppose, back at the stamp type of calculation. Like if you look at our herd, and it's about nearly 7 million in Ireland, You know, a 5.8% reduction would equate to about 400,000 animals. And so, just lastly, to answer the 3 questions that I've asked, you know, can the spectrum of activity of bacteriocins vary and be manipulated? Well, the answer to that is yes. And we see a great natural variation in bacteriocins. But a nice thing about bacteriocins is because they're gene encoded, it's almost limitless as to how we can change them. And we do quite a lot of protein engineering on bacteriocins. Can bacteriocins be used to modulate gut microbiome composition and functioning? Yes, niacin can survive intestinal transit and change the composition of the microbiome and its functioning, in this case by decreasing short-chain fatty acid analysis, which rebounds back afterwards. And if we think about this rebound back, it rebounds back fairly fast. So I think you could think of a lot of human applications for human infections where you might be able to use this. Can bacteriocins be used to cause a physiological effect? And all I can say here is this is very preliminary work that we're doing. We're just getting ready for submission at the moment. But feed containing bacteriocin-producing cultures was associated with rumen methane reduction. So I'd like to thank there was quite a lot of people involved in this work, principally Colin Hill and Catherine Stanton, who co-led these projects, and Pat Dillon on the actual animal, on the cattle side. And particularly Catherine O'Reilly, also on the, who did a lot of the niacin work. So thank you very much.
Thank you. Thank you very much, Paul, for your nice presentation. Very interesting to know such a wide range of applications for bacteriocins and the bacteria-producing bacteriocins. So we have time for a couple of questions from the audience. Is there any question? Can see very well.
There's a question down here.
Here, just behind the...
Thank you. Thank you, Paul, and congratulations. It was a great talk. I enjoyed it very much. And going back to the results you got with niacin, I was a little bit afraid seeing the results because I don't want to kill my bifidobacteria, I don't want to increase my proteobacteria, and I absolutely don't want to decrease my short-chain fatty acids.
Yes.
But it's a great proof of concept of the activity of, and survival of such a molecule. But do you expect to use other bacteriocins or modify this one as to increase bifidos and lactobacilli and preserve the synthesis of butyric acid in the gut?
Well, remember that, you know, there's a lot of nasty organisms like C. diff and C. perfringens, VRE, streptococci, that you don't like hanging around your system for too long. And so I think niacin would have, I think it opens up that niacin could have therapeutic uses for these. Yes, I think the fact that it rebounds so fast is good. I think also the amount of niacin that's used in foods was far lower. Like we're using 150 milligrams per kg for the animals here, so it's quite a lot. And this is pure niacin, not niacin. And so, but I do think it raises some questions. You know, in the first talk, the EFSA talk, you know, on the first day, it's something we're doing as well, is looking at chemical preservatives and their effect on the microbiome. And I think that, you know, they're probably subtle, you know, at the levels that they're used. But some organisms, like as you say, bifidobacteria, are extremely sensitive.
Thanks.
There is another question up there.
Thank you very much for the nice talk. 2 questions. One related to the silage test trial. Your controls were normally produced silage.
Yes.
To my best knowledge, there's also a lactobacillus fermentation in normal. Did you characterize that so that it's possible to...
Yes, we did. We characterized that. So what we did is we did like MRS counts on the, like we did lactobacillus counts, we full and microbiome of the silage that was made to feed the animals. And so yes, you're right, but the levels of them are far lower than the inoculated one. And they're probably not bacteriocin producing. I don't think we checked them if they are bacteriocin producing.
But it could be that the effect you have is, well, if the control also has an effect, then the effect you measure would be less than what it is in reality.
That's right. But I suppose we're comparing it to the control. The grass is kind of the same, and so like we're looking for a reduction over the control.
Next question. One way of modulating biomes could be to use the nutrients as prebiotics. Is that a concept that you have been working with? The way you feed a biome could influence what it is?
Yes, of course. Yeah, yes, like a prebiotic. Are you saying it in the context of the methane reduction, or are you saying it just in general?
In general.
Oh yes, yeah, absolutely. That's something that we work on. But when you do that, like what you're doing is, say if you use complex carbohydrates, you're enriching for organisms that have pathways that can catabolize those nutrients and organisms that maybe can be influenced by those organisms. And so yeah, that's another way, a key way to influence microbiomes.
Okay, thank you very much. I think we are running time and it's time for the next session. So I would like to thank you again, and don't move because I have a present for you in behalf of